Hello, can you hear me now? Can you hear me now?
Yes, yes, it's better now.
Okay. So I was saying that one of the most common use of PubChem is to download plant bioactive compound. Is to just to do your normal download. That is what we use it for commonly. But I'm going to explain further uses of PubChem. So I just downloaded a journal. The journal is talk about the chemical characterization. So if I want to make use of this journal, I'll quickly look at the GCMS results to see. Okay, so this is the GCMS results. While you are downloading your journal, your compounds, you're going to take note of the plant parts. The plant parts. So if you are working on a plant, don't just download all the compounds together. Separate them based on plant parts. Like this work is phytoconstituent identified in methanol extract of Comolena by GCMS. So in this table, they do not identify if it is the leaf or the stem. So you can quickly go to the materials and method, the sample collections, to know the plant parts which was used in this study. So let me see. Okay, quickly look at sample collection. Let me look at their sample collection so that we know the plant parts that was used. The reason why we must take note of the plant parts is for replication. Okay, the healthy leaves. So the healthy leaves of Comolena odorata. So they use the leaves. So that means after downloading this compound, I will save it as leaves. I will save it as leaves of Comolena odorata. So let me quickly look at those bioactive compounds. So these are the bioactive compounds under the leaf. So why I will just copy this. If I copy this, I will come to my PubChem and then I will paste in PubChem. So once I paste in PubChem, I'm going to download it. So one of the use of this PubChem is to download your structure. And please, meanwhile, let me show you some trick. Don't just be in haste to download. While downloading, always look at this. Please, if your mic is on, help me and put it off. I wouldn't know where the interference is coming from. Chinelo, your mic is on. Alright. So what I'll just do is, before I download, please look at this MF, which is the molecular formula. Anyone that has silicon, any structure that has silicon, please don't download it. SI, any compound that has SI, please don't download it, because SI is not recognized in Autodoc. The software we'll be using for docking does not recognize compound that contains silicon. So when there is any compound that has silicon, the software will not be able to recognize it. So what you need to do is, please don't download the compound that has silicon. So before you go to download, please look at this place. This is containing CH and oxygen. So assuming it has a silicon, I will not download it, okay? Because it will interfere with your docking. The pyrex we are using operate based on Autodoc, and Autodoc does not recognize some element like silicon. so don't download it. So what you just need to do is to click on this place, this code, this CID. You click on the CID. Once you click on the CID, then the next thing you will do is to click on download, because we want to download it. I'm just giving the first use. You click on download. So once you click on download, our interest is on the 3D conformer, but not all compounds comes with 3D. Some structures in this PubChem are in 2D. So some of them, when you open it, you will not see they are 3D, you will see 2D. But anyone that has 3D, then you click on this save, because we'll be using SDF. SDF stands for structural data files. So we shall be working with the SDF. So what you just need to do is to click on save, this place, save. Once you click on it, it will download the structure. It will download the structure, and it's showing you my system downloaded. Now it will not download with the name, but it will download with the CID. So every compound here has a CID code. So what we just did now is to download. You can also come back, copy the second one, and go to PubChem, and you go back by clicking on this capital C. It will take you back, and then you also paste, and then you press enter to download this structure. I think this is synonyms. This one here are different synonyms, so you just pick one of them. Let me pick caprinic acid, so I'll just separate it. Caprinic acid. So that caprinic acid will come up. You pick the caprinic acid. You check there is no SI, so you click on this code, on the CID code. When you click on the CID code, you click on download. So once it opens, you click on download. So we use this PubChem number one to download exact match. I've already explained about different kinds of search. I talked about the exact match. I explained about the similarity search. I also explained about the superstructure search. Okay, so these are I explained for exact match, similar structure, superstructure. Okay, so these are the major search. So here you come and click in SDF. So this is what you will do now to get the data, the structural data files, which you can use for your docking. Previously we learned how to model the structure. Now there are some chemical structures, you may need to download their structure, and you find out that the structures are not found in PubChem. So let's say for instance, I want to download this structure, and this structure, when I come to my PubChem, for instance, I want to assume that this structure is not found in PubChem, because PubChem does not give you a structure not found in PubChem, because it was by deposited structure. So if, for instance, this structure was not found in PubChem, they will tell me, okay, glory to God, I don't even know. This structure is not found in PubChem. This is 4-hydroxy, 2-methoxy, benaldehyde. This should be benzaldehyde. So you check the name. This should be benzaldehyde. benzaldehyde. So let's check properly. Benzaldehyde. Okay, so we see that most time, like the one I copied, the name was not correct, so you have to like correct the name. But let's assume that this structure was not found in our PubChem. The structure was not found in our PubChem. If you notice that the structure is not found in PubChem, remember PubChem is working with what has been deposited inside it. So if the structure was not found in PubChem, what you just need to do is to open your chem drawer, open chem drawer, and then go for convert name to structure. So that's what we'll do. You open your chem drawer. So that name that could not be found in PubChem, you open the chem drawer. So my chem drawer is opening. Once you open the chem drawer, you paste the name under your chem drawer and ask the chem drawer to convert the name to structure. Sorry, my chem drawer is actually opening. So you take the chem drawer to convert the name to structure. We have already done this part, so we can do it. I just want to show you that not all compounds may be found in the PubChem. You can only get those compounds that has been deposited in PubChem. So if it could not be found, you can also try other database, but the best is to use your chem drawer and convert the structure, the name to structure. Okay, the chem drawer is taking time to load. When that loads, will continue. So let me just point out something. If you are doing a work, which some of us we're going to start a work now, we'll choose what area of project we want to do, because our project work will start right now. So some of you who may wish to work on phytochemicals, not even, I will want everybody who will do a work on phytochemical, because we're going to use it to build our database. So after today's class, we are going to download 50 journals. So each person will have a plant. So a team, each team will have 5 plants, and that plant you're going to download 50 journals for each plant. Download 50 journals for each plant, and those 50 journals we talk about journals that has information on the bioactive compounds. So not just anyhow journal, but journal that has information on the bioactive compounds of that plant. That is what we shall focus on. So this is how to use PubChem to do your download. Let me open back the chem drawer. Let me check back the chem drawer, because you are going to use it. So I've already copied the name. What I just need to do is I will come to structure, then I will say convert name to structure. So that name that I could not see in PubChem, I will paste it in my chem drawer, and I will say OK. So chem drawer will be able to, a structure cannot be generated. I think we'll check if the name is correct. Okay, so this is actually the structure. This is the structure of that compound. We assume that we are unable to see it in our PubChem, because we don't want to leave any compound behind except the one that has silicon. So we don't leave any compound behind. So this is what we are going to do. Then you know what to do from here. Since we don't have the structure and we have checked our PubChem and PubChem does not have it, so we came to ChemDraw, and ChemDraw was able to generate the structure. So from here you do your modeling, you copy the structure to your 3D, then you model and you optimize from the 3D, then you save as SDF. So that is one way you will do if the structure is not found in PubChem. Alright, before I move further, do we have a question? Do we have any question?
No question.
Alright. Okay.
For that, because I just coming in.
Okay. So, let me now quickly look out for what are the other things we can do with PubChem. Are we just going to use it to download structure? It's not only download structure. We use it for so many things. I will quickly explain them. Outside the loading of structure, one thing we can do with PubChem, I've explained about the similar structure search. So we come back to that when we enter QSAR. Now, you can use PubChem. Let me place that compound. I can use PubChem. Let me search for this compound now. So I will tell you other things we can do with PubChem. Now on this place, once you click on similar search, I've explained about identity, that we have one compound. Then there are more than 1,000 compounds that has that similar search. If you want to see all of them, you click on this. It will open you up to all the compounds, and we have about 1,229 compounds under similar search. So you can get my video on similar search so that you understand what I actually mean by that similar search. I have done justice to that. Then we have substructure. This is where we always pay attention when we are dealing on structural activity. Maybe, like I always say, that compounds with similar structure, we have similar activity. So there are more than 1,000 structures that have the same structural pattern of this compound, but the difference is change in substituent, but it will maintain the same structure, but it will just be adding different substituent to it. So if I want to see, okay, quickly let me explain that with my aspirin. If I have aspirin, let's say I have aspirin, and aspirin is medicinal. Aspirin is probably anti-diabetic, or aspirin is anti-inflammatory. Now, and I want to develop another drug that we have similar structure with aspirin, and that compound has not been known. That means I'll come to similar search. I want to download compounds that looks like aspirin and use them for my study. What I will do is quickly come to substructure, and I will click on search all. So it will search all the substructure. You will see that for all the substructures, they maintain the same diagram with aspirin, but the difference is just some functional group added to it. So here I can now get as much as possible. This place is showing me 21,528 compounds that has similar structure with aspirin. So now you see that it is my own design of work, because I might choose not to work with phytochemicals and I'm now working with already existing compounds. So I can use my PubChem to download those 21,528 compounds that has similar structure with aspirin. So if you look at this place, this is the aspirin, the first aspirin. Then if you come down, you see that this structure is similar. The difference is that this is the aspirin. They only added, what they did was to add this ring, this place. So they removed the methyl and added this benzene ring. So you see that it's still maintaining this stuff. This whole part is from the aspirin. So they just removed the methyl and added this part. So they still maintain that same aspirin scaffold. The same here too. This is aspirin. This is aspirin. But what happened here is that the H here, the H of the carboxylic acid group was now replaced with another group, but still maintaining the aspirin structure. So that is what we mean by substructures, where you get structures that has the same structure with your aspirin, but the difference is they are substituents.
The screen is no longer showing.
Okay, let me check if my
It may be your network is showing.
Is it showing? My screen is showing. All right. Are you seeing it now?
Yeah.
Okay.
Yes.
So now you will see
Yes.
the same thing happens here too. This is my
Yes, I can see.
all right. This is my normal aspirin. This is the normal aspirin, but they now attached trifluoromethyl. So they are attached trifluoromethyl to one of the chain of benzene. So when you search for substructures, you are getting structures that have the same scaffold with aspirin, but difference in substituents. So I can use all of them and target the same target for aspirin. Since aspirin is anti-inflammatory, I can conduct anti-inflammatory on all those similar structures. I may end up seeing some of them that might even be better than aspirin, which has not been discovered as a drug. So that is one way of designing a structure, depending on what you want to do, depending on what is in your mind. So you can get structures similar to whatever you want to do from here. Then you can download all these. You can download one by one, but since I want to work with substructures of aspirin, I can download all of them at once by just clicking on download. Once you click on this download, because I'm already on substructures, I'm already on substructure. So once I click on this download, it will ask me what do I want to download. Remember, we have 21,528 compounds. So click on download, and then you come down. Okay, let me shift a bit. Okay, download. Then I want to choose SDF, but I will change this to 3D. So change it to 3D. Leave here as number 1 conformer. Leave it as 1 conformer. Change that to 3D, and then click on SDF. So once you click on SDF, it will begin to download that your compound. It will take time to download, and it will all download as a single file. So Later, I will teach you how to compress all your structures into a single file. So this single file now contains the 21,000 and thereabout compounds that are similar to aspirin. So it will take you time to actually download, but I don't need that, so let me just cancel it. So depending on your approach, we don't have business with superstructures. Now, similarity are compounds that look like aspirin. They may not have, they must not have that aspirin scaffold, but they look like aspirin. That's what we mean by similarity search. So click on select all, search for all. It will give you how many structures that look like aspirin. It may not necessarily be aspirin, but it will look like aspirin. Like this case, this is aspirin. This is methyl salicylate. You see under substructure. This one too under. Now look at this man now. This man is phenyl salicylate. It is similar to aspirin. When you look at this one is ethyl salicylate. Is not really aspirin now, because it's not maintaining that aspirin nature. So, but it's similar to aspirin. This one too is not maintaining that aspirin nature, but it's similar to aspirin. So we'll just be getting structures which are similar to aspirin, both the ones that looks like aspirin and the one that does not look like aspirin. It comes out under similarity search, and there are about 6,809 similarity search. So what we are going to now do is okay. Now, apart from downloading, also, like I'm seeing 16, what else can we do? Still on this aspirin, you will notice that if I go back to my aspirin, the initial place before I click on the, okay, let me come back to aspirin. Let me show you another thing we'll be doing with PubChem. But basically what people use it for is drawing of structure. I will show you. You can use aspirin. Let it open. Good. Now look at this place, this part. I'm done with informations under similar structure. You can check on related record. Related record may not be of importance to us. There is no much information on that related record. But the next thing I want to show you now is, okay, but let's see what opens up under the related record. But I know it will not be of importance to us. So I'm going to show you other thing you can do with PubChem right now. Let me quickly go back, because there's no vital information under the related record, though you can see if you can try that yourself. Now look at what is here. Look at these informations here: compounds, substance, pathways, bioassays, literatures, and patents. So I'm going to explain what these things are, right? Because we need to know what they are. We are not just downloading structure, but here it's telling me that there are 133 chemical names, chemical compounds that share the same name with aspirin. This means that this aspirin, maybe compounds that aspirin is attached to their names. So there are 133 compounds that has this title aspirin in their nomenclature. Here is aspirin, cation. This is a cation particles with a diaspirin. So here is butyl aspirin, caffeine. compounds that has aspirin under as part of their name. That is what is coming up on that compound. So under this compound that we have 133 compounds that has the aspirin as part of their name. You can come to this place and download them. You can also download those 133 compounds by choosing 3D and then click on SDF. The next is substance. So I'm going to explain to you what this substance is all about. Now, it's showing us some molecules that contains aspirin. Now, if you look at this substance, you are seeing two things: substance SID and compound CID. And all the CID all contain the same code 2244, which is all aspirin. They are all aspirin, but the substance ID is different. The substance here are constituents that contains aspirin, literatures that has reported aspirin, but that is not before us. There is no much thing to do there. The one that is of interest is pathway. Very important to us, to those in biochemical studies. What are the pathways of this my compound? So you from here and know the various pathways, of your aspirin. But then you must be careful of the organism you want to study the pathway. This is for domestic cattle, so you just have to look out for humans. Let's see if we can try to filter it to select humans. That is Homo sapiens. Okay, so you will just try to look out for Homo sapiens. This will tell you the metabolic pathways, but you be careful of the organism. So if you are interested, okay, this is for human. So this is biosynthesis of aspirin trigger this series. So if I click on this, it will give me informations on the metabolic pathways of aspirin in the human body. So if you study this place, it will provide informations on the pathways, the genes. Okay, look at the interaction. Let's look at the interaction. This place, once it opens, you provide informations on the metabolic interactions of aspirin, informations on the proteins of aspirin, and the genes, the genes that are expressed in aspirin. So you can use this. Okay, these are the targets: TPN. These are different targets, so different interactions. If you can understand them, it's just what you want to do. It all depends on what you want to do. Then these are some proteins that aspirin can target. Some proteins that aspirin can target. Some glycoproteins. These are some genes, the genes that aspirin can target. So you can go through this place to look out for some informations about the pathway. And if you don't want to, outside the pathways, you can check out for the bioassays. Very important. This bioassay is about the lab work, some lab work that has been conducted, different work, different publications that has been done on aspirin and their results. So this is percentage of aspirin formed during hydrolysis in undiluted human plasma. So here you'll be seeing different laboratory work, that is bioassay, different in vitro and in vivo studies that has been conducted on aspirin. So maybe if you are working on a particular drug, you can come here and study literatures on that drug, bioassays, not just ordinary literatures, but these people has a result of laboratory work and their results. This is stability in human plasma assessed as aspirin production. So you see different of those work. Metabolic stability in 50% human plasma assessed as aspirin release. So there are different publications, and it will be telling you the source, the data source, so you can quickly, okay, let's click on this one, anyone. So you can tell literature reviews from here, literatures on bioassays on your particular compound. I'm just using aspirin now as a compound. I've done explaining about downloading structure. So please, I hope our time is not off. Hello. Hope we still have time. We have some minutes left. Hello. Okay, we have 8 minutes left. So I'll quickly explain about these bioassays and we shall fall back to the next session. So this is the work conducted, different laboratory work